talk, which is going to be one of the 3 very exciting talks. It's going to be given by Dr. Simon Cole, who's the founder and CEO of Layton Labs, a frontier AI company working towards turning drug discovery into an engineering discipline. Layton Labs' mission is to transform every pharma and biotech company into an AI-native organization, compressing discovery timelines from years to weeks and multiplying portfolio productivity. This is a great problem to work on. Layton Labs developed Layton X and Layton X2 generative models that design lab validated antibody and peptide candidates with hit rates and binding affinities matching or exceeding traditional methods that screen millions of compounds. It's a huge problem in drug design. Most recently, the company has released Layton Wire, the world's first autonomous biologist design AI agent. Simon previously founded and co-led DeepMind's protein design team and was a senior research scientist on the Nobel Prize-winning AlphaFold project together with Demis and was invited in the Nobel ceremony that Jeff Hinton and Demis got the Nobel Prize last year. He's building towards a future where drug design runs autonomously with AI systems, designing, testing, and iterating on candidates at unprecedented speed. Simon is a vision guy. He was trained in vision. He has many CVPR papers. And he's going to associate now how he transitioned from vision to this exciting problem using machine learning and AI. So, Simon.
Thank you very much, Dimitris. Amazing. It's fantastic to be here today speaking to you. I will tell you a little bit about how at Layton Labs we're working towards programmable biology, particularly through the use of and development of generative AI for molecular design. So, as Dimitris said, I'm the founder and CEO there, and I'll walk you through the trajectory and future steps. So, before doing that, I really want us all to be on the same page as to how drugs work, how the current drug discovery process works, and then how we may sort of replace parts of that or aid in parts of that through generative AI, through the use of agents. So, like I said, let's first get the basics down so we're all on the same page. So I'll do that by walking us through some examples and give you a sense of how they mechanistically work as a drug. So first there's semaglutide, which also is better known as Ozempic. Of course, a huge success, something that is largely talked about for its success in treating type 2 diabetes and obesity. It's really a synthetic peptide drug. You can see it here depicted structurally. It's the helical peptide shown in this purple-blue tone, and it binds the GLP-1 receptor and activates it, and that's its mechanistic interaction with this target. You'll see this sort of depiction like this more throughout the deck, where we typically show the target in gray. Then there's another one I want to show you. It's caplacizumab. It's a llama-derived antibody. It blocks interactions between blood platelets that lead to blood clotting disorder. And so again, the mechanism here is binding and blocking in this case. And you can see the nanobody here shown in, again, a purple tone, binding and blocking to this blood clotting target in gray. Then there's another famous one in the industry called trastuzumab. It's a humanized antibody fragment that blocks HER2, and in doing so, treats breast cancer and gastric cancer. And HER2 is really a receptor present on tumor cells. And again, the interaction here is one of binding and blocking. And so, in a nutshell, what I want you to keep in mind is that the mechanistic way of working for a drug really is by binding and often blocking. So with that in mind, how are drugs discovered today? What's the typical process like? And you might be aware that it's very lengthy, and so I'll talk you through this. It's typically a process that takes 10 or even exceeds 10 years. It all starts with the basic discovery of the molecule first. So there's several ways of doing this. I'm just going to highlight a few. So one is, like I said already, derived from llama immune system reactions. Another one is literally proverbially throwing molecules against the wall and seeing what sticks in the form of large screening libraries. And then a third is essentially taking from human blood draws and extracting from that existing antibodies from patients and survivors. And so when we have these starting points, and I'm highlighting particularly antibody starting points because that's going to be of importance in my talk, we then typically have to iterate on these. Those are starting points, but they're not drugs. And so that's an iterative, sequential process that takes a long time and is suboptimal today. Again, something that we hope to change with generative AI. But from here, in the typical process, you then have to prove to the FDA, the regulatory administration, of course, that your drug candidates are non-harmful in humans. And you do that typically in animal models, for instance, mice, and then you graduate that to non-human primates. And with that data in hand, you can go to the FDA and apply for what's called an IND, essentially a rubber stamp that you can do clinical trials. So that typically takes about 3 years. And once you got that, you can then start doing human clinical trials. So phase 1 typically is one that looks at safety alone. You're not even looking at whether the drug works, just does it work without side effects in the human bodies. Then in phase 2, you're looking at dosing. How much of the drug do you need to give for it to have efficacy? And in phase 3, you typically do a large trial in the end that shows all of it together with the dose you've determined. Is it efficacious? Does it sort of work safely in a human? And if all of that comes together through that sort of 10-year plus process, you might be so lucky that the FDA approves your drug and you can sort of sell it in the market. And sort of this is really the funnel you have to have in mind that we're trying to address and make faster. And so what's wrong with drug discovery? A lot of that has, of course, been said. There's many reviews. This is a famous one in Nature Reviews, Drug Discovery, addressing the problem or speaking about it, teasing it apart. And it all boils down to a few things. So one, it's sort of super slow. It's very expensive. Discovering a drug currently takes $2 billion plus, and it's really the case that 9 out of 10 drugs fail today. So you have this large funnel that you hope your drugs go through, and only 1 in 10 makes it in the end. And of course, all of that is interconnected, and I want to argue that it actually has to do with starting with the wrong molecule. And that's what we're trying to change at Layton Labs, where we want to make drugs from scratch that actually have a much higher chance of getting through the trials, are cheaper in doing so, faster in doing so. And so let's briefly go into some more detail how we get to a starting point today. So I already mentioned one is immunization. Here the industry literally injects antigens into animals like llamas, and there's even more exotic ways, for instance, sharks. And so pharma companies and other companies in the space literally run llama and shark farms where they then harvest and screen antibodies from the immune response of these animals. And of course, obviously, llamas aren't humans, and so you then have to go and immunize these molecules. They're just starting points. It's also super slow. It's very limited in control. You're not sure where you're going to bind in the end. It's not precise. The second way I want to highlight is large library screening. I already mentioned there's billions, sometimes even trillions-sized screens where people screen random compounds and seeing what sticks. And so in this case, no animal is needed, of course. That's an advance. And it's somewhat tunable. However, at the same time, it typically results in lower affinity. You have to mature it to be a better hit, and it's just costly and takes time. Again, not a lot of control in that process. And then there's B-cell mining, where you literally recover naturally matured antibodies from survivors of diseases, from patients, and you extract that from blood draws. That has the advantage that they're naturally matured. They are typically a good hit. They're strong. They're human antibody from the start. But of course, it's very donor dependent. It's not scalable. You never have that for any disease that you want to treat. And so it has all sorts of problems. It's low in throughput, and also on top of that, it's complicated. It has ethical and access constraints. And so what we would like to do, the vision really is, let's imagine a world where we can design drugs for all diseases but instantly, and doing that with generative models. So, along comes a target associated with a disease pathway. We would like to be able to essentially prompt our model with that, build a model that can take that as input, and then makes drugs that work instantaneously. And sort of that, as I mentioned, involves, of course, binding, being highly specific, but then on top of that, have a lot of properties relating, for instance, to manufacturability and whether it works in a human. And that's the big vision. We would like to have that for anything that comes along. And let me sort of wrap up the intro by commenting on where we are as a field in sort of computational drug design. And I want to argue that we've made huge progress in just the last 5 years. It's really fantastic, actually, and in some ways reminiscent of the capability increases that we see in large language models. And so let's look at a timeline like this from 2020, which you'll see in a second why I picked that date, to 2026. In 2020, what happened was AlphaFold 2 came out, which of course is this computational model for protein structure prediction. And that was really sort of a seeding point for the field, I want to argue, like something akin to a breakthrough that all the work that followed really builds on top of in many ways. Then, a few years later, the first generative models came around for protein design, for unconditional protein design, where the goal was really just to make some protein that is not found in nature, that works in the lab. And models such as RF diffusion and protein MPNN from the Baker lab are prime examples for that type of model. Then shortly after, in 2024, AlphaFold 3 was released. That's a successor model to the AlphaFold 2 model, of course. It's one that has additional capabilities. For instance, it can model protein-protein interactions, something really important for drug design, because that's exactly what we want to model and design for. Then there was a model called AlphaProteo, also released from DeepMind, actually from the team I co-led at DeepMind, for protein binder design. And shortly after, at Layton Labs, we launched Layton X1, which I'm going to go into some detail in just a second. And then, in the course of the last 12 months, we've had a number of breakthroughs for antibody design. And the reason why antibody design is separated out here is that you can think of this as something like a holy grail in this area. It's actually much harder than making general binders. And on top of that, it directly addresses a key drug class that drug discovery companies care about. There was Layton X2, which was from Layton Labs. There was Chai2 from Chai Discovery, Cham from Nabla Bio, and actually quite a few others. This list is not trying to be comprehensive, to be honest. It's just trying to show you the trajectory of the space. And then recently, about 2 months ago, at Layton Labs, we launched the first agentic design agent that we also lab validated, called Layton Wire. And you might have already guessed it, I'm setting this up as an exponential sort of acceleration, where literally that's how I look at it. I think over the course of just 5 to 6 years, we've gone from everything was done purely in the lab to agents running antibody design. I'm going to use this slide as well to essentially give you a bit of an agenda of how I'm going to go through this talk. So I'm going to focus on the models that I've personally contributed to. So AlphaFold 2 as the root node, if you will, then Layton X1, Layton X2, and the antibody design agent, Layton Wire. So let's go through it in sort of this order, really. So with the intro in terms of modeling out of the way, I also want to make sure we all are on the same page as to what we're trying to design, what even proteins are. So let me give you a brief primer on proteins from here and why they're such fascinating object in and of itself, where even from a theoretical perspective, it's an amazing area to work in. So first, what are proteins? They're molecular machines essential to life. And I mean human life, but really any life. We don't know any other life forms on Earth or anywhere else that don't rely on proteins as the basic building blocks. So they consist of chains of amino acids that fold into a 3D structure. From a computer science perspective, you can think of this as a string formulated over an alphabet of 20 letters. And each letter actually corresponds to an amino acid. Now, they're folding up in space. I'm showing here in gray a particular structure that corresponds to the sequence below it. And really, every amino acid here comes with essentially a collection of atoms that designate specific bonds and specific biochemistry. So really, if you look at it like this, that's what's called the backbone. It's connected in space, it forms a particular fold, but looking at it at atomistic detail, it actually has a lot more granularity, and you can see all the side chains of the amino acids here. Importantly as well is protein structure is extremely important for a protein's function. Structure really does determine function, which is the reason why structure determination was such a critical step, which we'll get to in a second. Now, let's briefly zoom into this some more. So these amino acids, as I said, are collections of atoms that make specific biochemistry. For instance, this is an arginine or an R, this is a serine or a C for short, and this is an alanine. And all of these have particular subgraphs that give rise to particular biochemistry. Important for folding, but also important for their function and interaction with other proteins. And so it's really important to get it right literally at the atomistic detail. And let me motivate this a little bit more. So proteins have this particular property that even a single mutation can sort of make them cease have their function. And I think to this community, it might be an accurate metaphor to say, that is as if a single wrong pixel could break an image. And of course, human vision is robust. That does not happen. When you mutate a pixel, if you will, the image is still coherent. But that's not the case with proteins. So I'm showing this here in the example of hen egg white, which of course, is a protein that probably many of us consume every week, actually. If you make a mutation at the core, here shown in orange, where if we mutate from lysine to alanine, so literally just one little switch, it comes undone. It loses its function, it loses its structure, it becomes a disordered protein. And, again, that's very different, I think, to vision. And actually, mutations like this are also often the leading cause for cancer. So it is a prime sort of problem in biology, if you will, and something that is interesting to call out, because you need this precision for the proteins to actually work in the real world. So you can't get a single amino acid wrong. Then let's talk about the protein folding, again, because I think this gave rise to AlphaFold, which is this root node of the space. So first, Anfinsen, in his acceptance speech of the Nobel Prize for Chemistry in 1972, essentially hypothesized that sequence fully determines structure. It's called the thermodynamic hypothesis, and it has been widely accepted. And after he formulated it, people started thinking, if that's true, shouldn't we be able to predict it? And so, visually depicting the problem is really given a sequence, which is much easier to obtain in nature. We can sequence proteins quite readily these days, at least. What is the protein's 3D molecular structure? And so that's all. That's the protein folding problem. We want to map from left to right. And ever since 1972, people started working on this computationally, which is actually exciting, right? Like it's a long-standing big problem that people started working on. And that's exactly what AlphaFold 2 solved, if you will. And I should say that AlphaFold really stands on the shoulders of giants in two ways. So one is there was a fantastic public data set that researchers had collated since literally, again, also the 70s. And they have actually made that publicly available. They essentially, as a community, agreed to collate that data online. And that was a key piece for AlphaFold. And then second of all, there was CASP, which is a biannual blind assessment for structure prediction methods. And it's been run since 32 years, every 2 years. And it's a fantastic thing where people held out proteins that were deposited to the PDB for 2 years. No one else, except for the researchers, saw them. And then the structure prediction methods were assessed on that. And really, here's a performance curve over sort of 2-3 decades. On the y-axis, we see GDT, which you can think of as a structural accuracy metric. So it ranges from 0 to 100. 100 is best. And you can see that over many years, actually decades, the performance was middling. 40% accuracy, doesn't let you skip experiment. You can even see a dip, and that's not because the performance got worse, but because the held-out test sets got harder. And then came AlphaFold 1 in CASP 13, and that was a real uptick in performance, but again, not an uptick that would let an experimentalist set aside their experiment. But then with AlphaFold 2, actually, we reached 92.4, I believe, GDT in CASP 14, and that was a real big moment for the area. The organizers of the competition recognized that it was a solution to the single-chain structure prediction problem. And 92.4 GDT was considered, rivaling experiment, essentially. Here's another way of looking at the results of CASP 14, where what I'm showing on the y-axis is C-alpha RMSD. So structurally, the root mean square deviation between the true structure and the predicted structure, whereby I mean experimental structure when I say true structure. And AlphaFold achieved 1 angstrom RMSD across the held-out set of targets, and the sidechain accuracy was only slightly worse. And you can see it had such a huge impact in the field because even the second best entry was at 3 RMSD, which is worlds apart. So that was really a huge difference, even to only the second method. And thereby, it was sort of seen as this sort of, unilateral, in a way, advance in the field. So, and that's also why, the Nobel Prize was awarded to Demis Hassabis and John Chumper, and then also David Baker on the right for his contributions to protein design. Let's take a little bit of a look at some of the proteins and what it really meant. So here's one example that was held out in CASP 14. It's a large RNA polymerase The ground truth is shown in green, the prediction is shown in blue, and really looking at it visually, you can hardly see a difference. And that's sort of the accuracy that was reached, and that was vastly different when that happened in 2020. If you zoom into the substructure of these structures, they're called domains. They're even better sometimes. So really strong agreement and really astounded the field that was possible now. And maybe the sort of multi-domain performance gave some hint that soon after methods like this, including AlphaFold 3, were going to solve multi-chain protein structure prediction as well. So let's work a little bit on how AlphaFold 2 works. I want to go into some detail because it has been such an influential architecture that many works since then actually have built on. So I want you all to understand it a bit more and get some intuition why this works and what was key here. So true to the problem, it starts with a single sequence that we want to predict the structure for. That then gives rise to a search in a genetic database. And that gives a second input to the model, which we call a multiple sequence alignment. And really what we're doing here is we're doing a lookup into a large genetic database for similar sequences. And similarity there typically means similar function, but from a different species. And what happens in evolution is when species differentiate, they often keep the same function of proteins, largely even the same structure, but there's mutations accumulating. And for key contacts in these structures, these mutations must occur in both of the contact points. That's really key. That's called co-evolution. And this co-evolutionary signal lets us, with AlphaFold, in a learned way, determine these contact points. And that's a key input to AlphaFold. So from this multiple sequence alignment, it can start to infer what's in touch in 3D space. Then, from this input sequence, or its representation, I should say, we perform an outer product that seeds a pairwise representation. And what we're doing here is essentially, as opposed to, say, the standard transformer architecture, where interactions are modeled by pure attention, there's a lot of attention in AlphaFold as well. But here, we're giving it sort of a scratch pad to model more explicitly representations between individual residues. And that has been shown to be really key. When you ablate that, the model gets a lot worse. So we give it a scratch pad here to explicitly model pairwise interactions. And then the third input to the model is structural templates. I will say that's not very important, actually. In the end, the model performs almost as good without any templates. And so it's just, you can look at it as another way of prompting the model when you want to constrain it to a particular space. Now, from these inputs, we build two tracks of representations. One is the MSA representation stack, and then the pair representation stack. And then we've come up with innovative ways of having these two representations interact. There is quite a bit of domain knowledge by way of inductive biases built into what we call an Evo former. It's sort of fundamentally based on attention. The former part of the name nods to that, but there's a lot more inductive bias in there. For instance, we baked in particular triangular updates that respect the triangular inequality to bake in some geometry in how the pair representation can interact and things like that. And then at the end of Evo former, when we arrive at these representations, we have another network that maps these representations into 3D space. And really that is in AlphaFold 2 formulated as rotations and translations, and so really again bakes in inductive biases from geometry, but it turns out we later found in AlphaFold 3 that's not so necessary, but it was done that way in AlphaFold 2. And so look at this as a model trained to extract the co-evolutionary signal from multiple sequence alignments to a 3D structure with often atomic accuracy. And accuracy itself actually leads me to the next slide, because we found a way, second next slide. First I want to highlight there's some other innovations in the model, for instance, recycling, where we feed back representations from the end of the model, and that also made a difference. There's actually many such things. I point you to the publication for more of this. Sometimes we thought of it as something like 10, 15 key ideas, all of which could be a large publication, but sort of crammed into one architecture. And then I was going to speak about confidence prediction. So that's something that I spend a lot of time on this architecture, where we have two ways of predicting confidence in AlphaFold. So one is a per residue confidence. That's something that I've built, and it turns out very well calibrated. So when the confidence is low, that typically results in, or that typically means at that point the prediction is poor compared to an experimental structure. And then we have a second confidence prediction, which relates to the pairwise alignment or pairwise, let's say, placement in space. And again, sort of really important, both of them now, also for drug design and binder design, where this is something where we're using AlphaFold to check how confident it is when we design binders. More of that in just a second. So with structure prediction, quote unquote, solved, how do we design drugs? And I think one way of looking at AlphaFold is as a computational microscope. It lets us predict the structures of proteins from their sequence, like I've defined it earlier, but it doesn't let us do that in a way where, we could generate proteins that don't exist in nature. It's sort of built like a microscope, but we want to go beyond nature. We want to make designs from scratch that adhere to particular requirements, that bind to unseen drug targets, and that's what we want to do in drug design. So how do we design binders and ultimately drugs? And we can formulate this as a conditional generation task. So again, starting from, say, a sequence and a structure of a target that's associated with a disease or a disease pathway, we want to map this to a binder that interacts with that target in a highly specific way, getting the biochemistry of the interaction just right at the atomic level, where, if you zoom in, you need to really get the hydrogen bonds right. You need to get all these interactions just right, hydrophobic interactions, that it specifically and with high affinity interacts exactly at the spot that you want to have it interact with. So this conditional generation, of course, is something that we have good handles on as a field, also in computer vision. And what we ended up doing at Layton Labs is really we treated it as a point cloud of atoms and then condition that on the target. Of course, there's many ways of how exactly you can do it, but that's the fundamental idea. So, if you represent your target in terms of its coordinates or any other representation, give that as conditioning, and then you initialize a binder essentially, from scratch or as a, Gaussian noise, depending on your model of choice. We are actually working with denoising diffusion models, which you're going to see in a second. You have this as input to your model. We're calling our models Layton X. So, again, target conditional, binder from scratch, and what you want is for the model to then spit out something like this, where conditioned on the target, we can make this binder from scratch. And what you can see here in this denoising diffusion trajectory of Layton X is that it really does start from raw noise, and then over time it sort of, determines and sculpts its predictions, its generations, into a well-formed protein with good secondary structure and with atoms appearing, side chains appearing that make these specific interactions with the target. And you might notice that there's some innovations in there where, for instance, we do not fix the target completely. We actually let the model co-design some of the side chains so as to make better bonds, similar to how it would work in nature. The side chains actually can move around, for instance. Then, as I set up the whole talk, AlphaFold 2 was this root node, and I want to draw a comparison to how predictors, generally speaking, as a development pattern, give rise and enable generative models. And I think that has been the case in computer vision as well, so I want to make that comparison for the community here, where in computer vision and image generative models, I want to argue that everything started with ImageNet. So, fantastic data set by Fei Li and others in 2009. And then very quickly, that gave rise to the ImageNet challenge, and it gave rise to accuracy metrics that the field then used to build predictors of. And these predictors, famously, of course, AlexNet in 2012, which was, the first breakthrough, and then quickly improvements were stacked on top, for instance, by Google's Inception v3. And that gave rise to the Inception score and the inception distance, which in turn allowed people to rapidly iterate on image and video generative networks, such as GANs, of course, and diffusion models. So this pattern from dataset to challenge and metric to predictor to generator has repeated itself in structural biology and protein design. So in that case, we had what I already mentioned, the protein data bank from 1970 onwards. Like I already said, literally researchers depositing their protein structures for anyone to use in this joint database online. Really fantastic resource. Then the CASP challenge, also already mentioned, started in 1994. Now we're at CASP 17 this year, and CASP 14 was, like I said, the one where AlphaFold 2 stood out. That gave rise to AlphaFold 2 in 2020, 2021 time, which is the equivalent, I think, of the Inception network in that it allows us now to rapidly make advances against this oracle, if you want to call it that. And that gives rise to generative models like RF diffusion, as shown here, and others. And really, I think the key thing is that these predictors let you rapidly work computationally and advance towards optimizing them, but equally in protein design, they are critical to actually sub-select and rank designs that we want to put into the lab. And I'm going to talk about that in just a second. So with that, let me introduce to you our first foundation model, Layton X1, that we released 12 months ago, pretty much. And I'm going to show you a denoising diffusion trajectory. And I think what's really cool here is that you can see it starts from noise, then rapidly builds out structure. It puts atoms in place, and as it sort of denoises this structure, it forms these hydrogen bonds, biochemical interactions, shown in these pink dashed lines, with the target, shown in gray. And I really love that trajectory, so I'm going to show it to you again to fully appreciate it, if you will. And what's so exciting here is that it's almost like a search over how to make bonds here, and that's learned. We don't constrain it to do that at all. It almost looks like a simulation, but really it's a rollout of the model over the trajectory. Then in this space, what's key is experimental validation. We cannot stop at assessing it against a computational predictor or oracle. We have to take one more big step. It really needs to work in the lab. So how do we go from prompt to lab? So it all starts with just that, a prompt of the sequence and structure of the target. We also specify where we want to bind. That's shown in orange here. That's important, because functionally speaking, you often want that precision. It's residue precise. It's even atomically precise in doing that. Then we generate binders, for instance, with Layton X1. We then go and rank them with a predictor, for instance, AlphaFold 2 or AlphaFold 3-like models that takes the form of essentially cross-checking, does the prediction agree with the designed bound complex, and how confident is the model? Bringing back that idea of well-calibrated confidence predictions coming in here and being really important. And then the last step is critical. We have to put it into the lab to see what's real and what isn't. And that's why at Layton Labs we have our own wet lab in San Francisco. There's many ways to validate binders and designs overall. What you see here is from our lab, essentially fluorescent imaging that is run on human cell lines. Here, kidney cells. Imagine, essentially kidney cells on a Petri dish. We have them express our designs alongside a fluorescent tag. That's what's shown in green. That means our designs are expressing well. And then we flow over it targets that have an orange dye. And when that orange dye, and with it the target, gets immobilized on these cells, that means binding has occurred. So you can see in this top right corner, that would be a great binder. And that's one way of assessing it. Again, I want to make just come across how critical it is to test in the lab, again, because even a single amino acid can make the binder or whatever function you're designing for break. So let me speak a little bit more about how we validate it. First on Layton X1. This has been a foundation model that was designed with the essence of foundation models in mind. It's not tuned to any particular modality for biologics, but it works across the board. And we've validated it for peptides and mini binders. And so peptides, and in this case cyclic peptides, are a drug class whose application it essentially could be to rival small molecules, particularly cyclic peptides, because they stand to be orally administered because of their cyclization. They literally form a ring, which is not normally the case with proteins. And we've tested our model for this particular application, and it worked really well. So here are some designs that we've published for cyclic peptide design with Layton X1. Again, you see these zoom-ins. It makes these highly specific interactions with the target, and that results in it working really well in the lab. We've seen success rates literally on all the targets tested here exceeding 90%, which was quite astonishing, to be honest with you. And the binding affinities were good as well. So this already today works quite well. It's for hit finding, that we have to go and get at other properties. I'll talk about that in just a second. But for hit finding, this works already very well today. Then let me talk about the mini binder application with Layton X1. So mini binders are proteins that sit somewhat in between in size peptides and the larger antibody class. They're sort of 80 to 120 amino acids long, and they're kind of undefined fold. They can take on quite diverse different folds, and you see a particular example here. They're interesting for potentially targeting agents, meaning cell therapies, gene therapies could be targeted to particular cell types with them, and then also research reagents. And again, we've made a lot of designs for Layton X1 that we put into the lab, and it worked really well in this case too. So we've considered targets that are benchmark targets in the community, and we, without any optimization, first time trying it, hit every target, bound to all of these different targets shown here, and did so with very strong binding affinities ranging down to what's called the picomolar range, which is a very tight binding that is really already close to what people would consider a drug-like, at least in terms of affinity. We've also benchmarked that at the time against other models that were out there 12 months ago, and it clearly exceeded in terms of affinity and hit rates in that respect. Then in December, we launched our second model called Layton X2. And the big advance here is that this model now also allows you to do zero-shot drug-like antibodies. And why that's important is, like I said earlier, antibodies are this major drug class that plays a huge role in approved drugs today and is still expanding. It's really of high interest to the drug discovery industry, and we've shown that we can make them from scratch against unseen targets with high affinities and across different antibody formats. So even within that class, there's different subtypes. So on the left, we're seeing a nanobody antibody. On the right, we're seeing what's called a single-chain variable fragment. And across these different formats, we're doing really well. We have strong affinity binders in the lab down to the nanomolar range for nanobodies, down to the picomolar range for single-chain variable fragments. And I'll get to what we mean by drug-like here as well. That was a key advance too. So not only are these hits, but we already see that they get good properties. And then that same model, because it's a foundation model again, also does cyclic peptides and mini binders. So we validated Layton X2 against the whole stack of targets. We've actually done many more since then, but this gives you a flavor. And we've picked them to be representative of diverse therapeutic areas. And what we showed by doing that is that the model really generalizes. It's not just working on one particular target or disease area. Particularly in the launch in December, we've picked targets that never had any antibody bound to them published structurally, so that we could make sure we can claim, this is not memorization, it actually works in a generalizable way. And we've since then also tested these antibodies in full monoclonal antibody format, in IgG format, so those are then larger molecules that you conjugate to a constant region. I'll spare you the details for now, but that's really what drug discovery companies care about, and we see that work really well as well. Then let me talk about drug likeness. So what's important is to go beyond just binding. It's sort of perhaps second order in a way, but it's really critical. So you want it to have low liabilities, these designs. You want these designs to be manufacturable. So it's actually, it could actually be an industrial project. Product, and you want them to be low polyreactive in that it shouldn't interact with other molecules in the human body. And so we've tested for thermostability, which is essentially the melting point of these molecules. You want that to be high. We've tested for monomericity. You want them to not self-interact, so you can, in sort of high density, ship them in vials, for instance. You want the yield to be high, so it's cheap to make them. Yield here meaning how well can a cell make them. You want polyreactivity to be low, so no off-targets, and you want them to be low in hydrophobicity, which means they're less likely to aggregate. And the exciting thing across all of that, sparing you again some of the detail, is that this is normally really hard to achieve. People sort of need to iterate on that sequentially. It's often a game of whack-a-mole. When you optimize one property, another property gets worse. And here, essentially, we, for 47% of the designs made with Layton X2, saw that we already clear important hurdles jointly without having this multi-target optimization. And we also directly compared against therapeutic antibodies here, shown in gray, and see that this really already looks good from the start. And so that speaks to that idea of ours of making generative models that can make sort of drug-like molecules from the start. We've also gone a step further. We've made antibodies and tested them for what's called ex vivo immunogenicity. So immunogenicity is whether the human body reacts adversely to these drugs. We can't do that in humans today. We don't have approval for that. That's why we did it ex vivo, which means we essentially collected blood draws from 10 healthy donors and then put our designed antibodies in those blood draws and looked at what the immune system does in that blood. And particularly, for instance, you can look at whether T cells proliferate, so essentially whether they divide, which would be indicative of a reaction. And you can look at whether cytokines get released. Those are hormones that signal within the immune system that the immune system is under attack, if you will. And excitingly, even though admittedly we didn't test that many antibodies, the ones that we tested had low ex vivo immunogenicity. That's only a first step. There's much more to be done. But again, I think that indicates that there's something there where we can get to good molecules from first generation. Then I want to talk a little bit more about cyclic peptides in the context of Layton X2 and how we've tested that here. So specifically, we've compared Layton X2 designs on 2 targets, PHD2 and KRAS, against a literally trillion-scale random library. And we picked these 2 targets because they were publications for exactly that screen on these targets. And excitingly, we saw that with only 10 designs, we can essentially beat that screen. We, on PHD2, got stronger binders by 2 more orders of magnitude stronger, testing only 10 designs compared to the trillion-scale screen. And we did that with 1 day computational work and then 4 weeks in the lab, compared to something like 3 to 6 months in the lab, a lot more human effort, a lot more reagents going into this. And I think that, shows how this sort of computational precision can replace brute force screening. And then the same thing on KRAS, which is historically considered undruggable. It has been drugged since, but it's sort of the poster child of difficult targets. And again, with just 10 designs, we found a binder that was as good as what the screen can identify. And then another exciting project done with an external partner, they have tested Layton X2 cyclic peptides to block and inhibit ion channels. Now, ion channels are protein pores that mediate ion flows across the cell membrane of neurons. And so that could really have large implications for pain medication. And it's considered really, really difficult. So here's an example. Here's the example that was used here. So in gray is the human NaF1.7 ion channel. It's a sodium channel that lets sodium ions across the pore. And in orange is the design that the collaborator made with Layton X2, where that's again a cyclic peptide. And they measured this in the lab and found it to block the ion channel function and ions going through. And again, that could be very exciting for pain medication potentially, and it hasn't been done before. Really, this has been among the hardest targets out there to get computationally, and so far people have actually relied on natural starting points, such as literally the venom of the green velvet tarantula shown here. And so again, a point that I want to make, where I think with methods such as these, with generative AI, we will be able to do this from scratch rather than being at the mercy of nature, at the mercy of search, and that's what's exciting about this. So before I conclude, I want to conclude this section on our models and how we validated them. So what I want you to take away is that they're frontier models. We formulate them in a way that is agnostic within biologics, so designing protein drugs. And we've done that in a way where we're modeling it at the atomic level. And that gives high specificity, high affinity, and gives us properties like low polyreactivity, essentially for free, because they're so specific. And that's the approach we've taken here. So we've then gone and put it a notch up, if you will, and created an agentic system around our models and tools. We called it Layton Wire, launched it 2 months ago. It's the first lab-validated antibody design agent. And we think of this as essentially a force multiplier for drug design teams, for drug discovery teams. I'll show you a brief video that condenses 5 hours of the agent running into 2 minutes. So it's going to be pretty sped up, but I'll talk over it, and you'll get a flavor of what it's able to do. So we're asking it here in the prompt box to make nanobodies that block human IL-6. And that's literally it. Just from this, the agent now starts running. It's asking questions back, wanting more specifics, but we essentially just tell it, you go and do it. We just want to be able to test nanobodies in the lab. And from here, the agent then goes and constructs a to-do list of all the tools that it wants to run and in what order. It has access to external databases, for instance, the PDB that I already mentioned. It goes and queries it to find target structures, analyzes these, which one might be best suited for this task. Then it calls sub-agents that run hotspot research, essentially where to bind in order to achieve the inhibition function. It does that structurally, reasons about that, essentially forms prompts for the Layton X2 models from that research, and then runs Layton X2 in the Layton Labs platform with different prompts across different projects, and then goes and analyzes that. We're going to see that visually in just a second. Here's the different prompts that it launched for Layton X2. It then analyzes that, learns from it, and can spawn another round and another round. And so it really does this sort of exploration, exploitation step like protein designers would do in practice. We know that internally and from people using it. And at the end, it conducts a QA, so a quality assurance step. Here are, by the way, some nanobodies designed by the agent here in the platform. It then goes in, does a quality assurance step, analyzing the sequences, clustering them, looking for sequence liabilities, and suggesting which one to take into the lab. And 5 hours condensed to 2 minutes in this video. However, from user studies, we know that this work, if done from scratch, even for a PhD protein designer, typically takes 50 times longer. So you have a 50x speed up. I think in engineering, whenever something gets 10x faster, something exciting is around the corner. And I think that's the case here as well, where we think we'll really be able to help teams expand the portfolio of things that they can do at the same time. So really being a force multiplier. Now, I promised we lab validated this, and those are some of the results. So literally from prompts like this, disrupt PRL's receptor interaction with nanobodies, we achieved very tight binding in the lab. Here's 3 prompt examples that we've published on, and all of that worked in the lab without any iteration, just from prompts like that. And then excitingly, we also asked it, given scientific literature, to identify the function described in that literature and make designs just from that. So essentially prompting with a PDF of a science or nature paper. We've done 20 such papers, and the model autonomously went and designed nanobodies in this case. One of these we've tested on the lower left-hand side. We've put designs of human transferrin 1, which is a blood-brain barrier target, into the lab, and 11 of 40 worked just from that prompt with just a PDF of the corresponding paper. Then I'm going to start to get into closing my talk, and I want to highlight some open problems, which, again, that's not comprehensive, there's plenty, but one of the standout ones is protein dynamics. So really what I've showed you is modeling essentially proteins statically. The denoising diffusion trajectories, they look dynamic, but really at the end, we're sort of modeling things in static state. And that's just not the case for nature. Really, proteins are these jiggly objects, and that can be very important for particular functions, for instance, enzymatic functions. This is an enzyme, by the way, that is shown here. Then, I mentioned that earlier, we also have no way today to reliably predict human drug response. What happens if we put that drug into a human? Very, very difficult, unclear what needs to happen for this to be solved. At the very least, I think we need to collect more data, and how to model it, that will be seen in the future. And then similarly, there's systems biology. So everything I've shown today was really modeling protein-protein interactions at the molecular level, target and binder. But really, these proteins, they of course don't live in isolation. This is a picture, an artistic rendition of a virus in a cell, and you can see it's really tightly packed. There's so many proteins around, there's huge interaction networks of proteins. Then if you zoom out, there's of course cellular systems, and zooming out again, there's whole organisms, and they're intensely complex. Given all the functions of an organism are given rise to by protein interactions, but we have no way today of modeling that at the cellular, let alone organism level. Then I thought it would be interesting to come back to this picture of the vast progress that I showed earlier over the last 5 years and do a few predictions, like what do I think this will look like for the next maybe 5 to 10 years. And maybe naturally, I'm an optimist in this space working on this, so it's going to be an optimistic take and one of abundance. But I do think we're on a fantastic trajectory. That's why this is what it is, in my view. So what does this look like over the next 5 to 10 years? I think what's what's going to naturally happen next is self-improving design systems. I think that follows from agentic systems that can either work with tools and better predictors, or with lab in the loop to rapidly improve the systems themselves. Then, I think we're going to make progress towards perhaps wholesale predictions or simulation, at the very least on the pathway level. Could again be done in agentic ways or otherwise, but something that definitely needs to happen, and I think it will. And then I think there's a lot of chance for personalized design, for instance, conditioned on the genome of a patient. That would be tremendous towards so-called N-of-1 therapies, for instance, for rare diseases. But also, if you think about it, the whole system for clinical trials really is population-based. But that doesn't mean you're sort of perfectly going to slot into the population that was tested on. So even outside of N-of-1 medicines, I think that would be tremendous. And then predicting human drug response. When we collect more data that is machine learning ready towards that, I'm optimistic that we're going to be able to learn that. And all of that together, I think, literally will allow us to essentially solve all disease. Now, that's really a vastly optimistic statement, but I think we'll get there. It's a question of time, not if, in my mind. And I wanted to close with essentially that message, that this is a really incredibly high impact area to work in. Then, finally, I think we're at an inflection point, and biology is finally becoming programmable. I wanted to thank my team at Layton Labs, who of course did most of this work. It's a really fantastic team. I was fortunate to bring others over from the AlphaFold team, from the DeepMind Gemini team, and then many other places across academia and industry. And we're spread across London and San Francisco. And so I'm telling you this because, if this piqued your interest, join us. We have a lot of roles open, both in London and San Francisco, member of technical staff roles, applied AI research roles, that we're trying to fill rapidly because the space is really going off. And then, beyond that, we're, of course, also having our models and agents available in the Layton Labs platform. If you want to give that a spin, apply for access. I would encourage you to do so. We really profit a lot from user feedback, making our systems better. And then with that, I'm closing close to on time. I want to thank you for your attention, thank the organizers for the invitation. Please reach out to us directly, follow us on X, either the company or me personally, and you can find our technical reports on our website and on the bioarchives. And I'm looking forward to your questions. Thank you very much.
Thank you, Simon, for an exciting talk, and I guess you can understand the complexity of the biology. And he's doing something that's futuristic and much needed. So we have time for like 2-3 questions. If somebody would like to come up to the microphone and
Hello. I have a probably naive question. My background is in psychology, so not professional at all. I'm just curious that, given that the input is the target that you need to bind to, what about the discovery of the target? Is that going to be the bottleneck now? Is there a way to speed it up? Thank you.
Yeah, that's a great question. That's sort of definitely the case that what people call target ID, target identification, is almost a research field in and of itself. Typically, we're not short of targets, to be honest. However, many diseases will likely profit from better targets. And so it is true that is a very exciting area that I also think actually models such as ours can aid with. When I was talking about research reagents at one point, that is also towards actually understanding target biology better, which sort of interactions in, say, a diseased pathway to block, and really make target identification faster and better. And so definitely something that is sort of, I think, downstream from what I've described today, but equally as exciting.
Hi, great talk. I'm curious with any of your wet lab work, if you're working on building or contributing to any of the cell imaging data sets and trying to potentially skip a level in your modeling to cell structure based on targets, or if you think that's just intractable.
No, I think that's very interesting. I think it's generally a data bottlenecked field right now. I think we've sort of squeezed a lot of juice out of the PDB and other data sets, and essentially what I think the air gap right now to the clinic is actually more data sets potentially of the likes that you're describing. I think the trouble is how to do that in a highly scalable way that essentially results in machine learning ready data sets. The field isn't really short in data in a way, but they're heterogeneous and sprinkled around and too small and not homogenized. And so, the kind of efforts that you're alluding to, I think we'd be very excited about seeing where they're going and potentially contributing.
So this is actually kind of relevant to the previous question on the wet lab. Seems like the agent stops there and you have to manually go and verify that. Is there work that you're doing on automating that, basically just putting that in the agent loop?
Yes, yeah, great question. Exactly, sort of this self-improving idea relates to, I think, what you're saying. There today is already some experimental service providers that essentially have APIs into their lab. And so, closing that gap, giving that API to an agent, I think that's super exciting. There are many complexities around it that are probably obvious. No single lab will cover the entire space. Unfortunately, even though it's a compelling idea, labs typically are not as modular as you would hope, in that you can't just test anything and rapidly behind the lab API you could sort of rearrange that. However, for more common workflows, I think that's a very tractable thing that we're already looking into.
Dr. Cole, you showed that in addition to antibodies, you can also design much simpler mini binders and peptides. Do you foresee or do you expect those much simpler mini binders, peptides to be able to replace antibodies as drugs?
Yeah, that's a great question. I think potentially yes. The motivation for doing that is probably the key piece, which perhaps is implicit in your question. So antibodies, typically they are administered intravenously. They don't survive our gut biochemistry. They get chopped up. And so that's why small molecules are still a very interesting modality. They can go through our gut and then into our bloodstream and get distributed into our body without requiring intravenous administration. And so for that very reason, I think peptides, in particular cyclic peptides that I showed, are very interesting. And so once we are able to model human drug responses, I do think there's nothing in the way of also designing mini binders or peptides that are non-immunogenic, that are non-toxic. I think it's just today the starting point that human protein scaffolds provide. I didn't talk about that a lot, but that's essentially the base motivation for antibodies. They give you a leg up. They provide you with a better starting point than any other modality, which is why they're so popular. And so, to just close the answer, I think the answer is yes, once we have a better handle on, human drug responses.
Thank you.
So the next step for drug design was toxicity prediction. So wanted to see if Gen AI can actually help in at least predicting the in vitro toxicity, if not in vivo, because of systems biology.
I think it will. I think it's a data set problem. That's what's stopping us. The interaction networks of the human body are so vast and largely, still I would say it's accurate, not understood, that from scratch or in any sort of theoretical way, that's impossible today. So if we have enough data that allows us to build predictors, then yes. So in some ways, it's maybe the boring answer, but we do need to probe the system more to then build a generalizable predictor. And then I think it will be possible.
So can high throughput screening actually help? It's being used all over to actually predict in vitro toxicity a lot.
It could, to my knowledge, it's still not at the standard where you can build a predictor of these systems. Maybe it's not at the right size. Perhaps you have visibility of that research more than me. I think I would sort of probably point to an existence kind of proof argument, which is molecules today still are often toxic, so clearly we haven't got it under control yet. But I do agree, collecting more data there and then starting to predict it with reliability will be the way forward.
Thanks.
So you have identified that protein dynamics is one of the open challenges, and I was curious, what are your initial thoughts in tackling this problem, and what makes it so challenging?
Yeah, great question. So, there has been a long-standing field of molecular dynamics, which essentially came up with physical functionals, sometimes fit to data, but not a lot of parameters that were used in MD trajectories. And on some sort of limited number of systems, that works well, but it doesn't generalize well. And so what people are doing since some time now, or still fairly recently, is building molecular force, machine learning force fields to try to improve that. And I think that's starting to look quite exciting. There has been quite a bit of promise and progress on that. And I think part of the reason is the brittleness of classic MD force fields. So maybe machine learning will come to the rescue there as well. But beyond that, the problem, I think, is twofold. It's sort of the accuracy of these force fields, and on top of that, the sheer compute that you require. So I think maybe Jensen and NVIDIA will solve that for us over time, or other providers in this area. But today, to model anything at sufficient either time scale or atomistic scale is pretty impossible, which doesn't mean it's not going to be solved, but it remains difficult today, even computationally.
Thank you.
Hi, thank you for a very, very good talk. My question is about this transition step from going from the silicon to the wet lab. So you could generate like millions of sequences that could be viable. How do you make the selection? What metrics in particular do you think are very useful in selecting the best ones, because you can't test all of them?
Yes. I think there's maybe two answers to that. So one is the generative model themselves. In our case, Layton X1 and X2, they have to be good. Like, if their success rate, even unfiltered, is too low, there's probably nothing that at least filter currently couldn't do. So you have to start with a strong generative model today. And then, on top of that, we're using structure prediction models, where I briefly alluded to that. We look at sort of when you fold that design in complex, does the structure agree? On top of that, the confidence metrics. And the particular thresholds and metrics that we're using, I'd invite you to look at our technical reports. We actually report them, but they're essentially thresholds on confidence metrics and structural agreement. And then that together lets us achieve success rates in the lab that allow us to test only very few designs per target. I think I briefly mentioned none of the targets we've gone after we've ever made more than 100 designs against. And that's only possible because the generator and the scorer are both strong.
Thank you.
Please be very brief, okay?
Yeah, thank you. My question is, like you mentioned, the data in this domain is kind of scarce. And so that requires kind of domain-specific model design, like AlphaFold 2 or 3. I'm wondering, like, do you envision like this trend will be last for a long time, or the field has reached a turning point where there will be a vast amount of like other general data that can help to build a foundation model, like that bypassing this specialized model design?
Yeah, no, I think there's a bit of a rush towards larger data sets and expand data. Something I didn't touch on that's really fruitful in this area, and I know has also been something that was done in computer vision before, and actually was part of AlphaFold 2 as well, is synthetic data. So maybe it's not so widely appreciated, but even in AlphaFold 2, the ratio of synthetic data to lab data was 3-to-1. So we used more synthetic data, self-predictions distilled back to tune that model. And I think that's something that's really fruitful in this area as well. And then just briefly, in terms of the specialized architectures, actually that's starting to fall. So even within our work, we're incorporating much less inductive biases today. It's actually becoming less relevant as you get more data, as we know. But it's definitely already the case that you need much less equivariance is baked in, let's say. It becomes more general systems.
Thank you.
Hi, thanks so much for the talk. My question is about this notion in the field of out-of-distribution sampling, or guiding these models towards samples maybe outside their training distribution. My question is how you think the field should best approach this problem, or whether you think there's a fundamental limitation in the way these models are trained.
Yeah, I think honestly, it's not as big a problem as I initially feared. Like in the beginning, some of the models out there, they were quite out of distribution. For instance, there's in the field well-known models that produce very highly charged proteins with high charge amounts on the surface, for instance, which you don't find in nature. They actually, interestingly, still express well and things like that, but they have other properties that are not, for instance, conducive to drug design. So there definitely have been biased methods out there, but it doesn't seem to be a huge problem, honestly. And I think what helps with that is, in some ways, you can think of the generative models being a fantastic prior over, what would be an appropriate binder, and then the scorer narrows that down further, maybe cutting off some tails that are a bit far away. That's a hypothesis, by the way, but my gut feeling is it's not a huge problem.
本演讲实录已收录至雷锋网 CVPR 2026深度专区，现已全面上线。专区不仅全面收录了重磅论文的工程化解读、专家前沿演讲，更将持续更新前方记者的第一手会议动态。扫描下方二维码或进入雷锋网 CVPR 专区即可查看。